it's Gabrielle, and this video is a sample from Studyclix Boost, our new rapid revision tool. Go to studyclix.ie/boost to find out more.
This video is on the very popular experiment, the isolation of DNA from a plant. When I do this experiment in school, I always use a kiwi, but you may have used an onion or another plant. What is interesting about this experiment is that, in the Leaving Cert Biology exam, you may be asked to explain the reason behind many of the steps in the experiment, and that's how I will go through it here. We peel the kiwi and chop it into small pieces. This increases the surface area and breaks down the cell walls. We then place the chopped kiwi into a mixture of 3 grams of salt, 10 centimetres cubed of washing up liquid, and 100 centimetres cubed of distilled water. The washing up liquid breaks down both the cell and nuclear membranes, allowing the DNA to leave the nucleus. The salt causes the DNA to clump together. We then place the beaker in a water bath at 60 degrees Celsius for about 15 minutes. This temperature denatures the enzymes that would normally break down or digest the DNA. We then place the beaker into an iced bath for about 5 minutes. This cools the mixture quickly, preventing any remaining enzymes from destroying the DNA. We pour the mixture into a blender and blend for 3 seconds. This further breaks down the cell walls. Any longer than 3 seconds would break down the DNA strands, and we don't want this. We then filter the mixture using coffee filter paper. The filtrate contains the DNA and the proteins. Coffee filter paper has larger pores than normal laboratory filter paper and so allows the DNA to pass through it. We then place 5 centimetres cubed of this filtrate into a boiling tube and then add 3 drops of protease enzyme. This enzyme breaks down the histone protein surrounding the DNA in the chromosomes. We then trickle 10 centimetres cubed of freezer-cold ethanol carefully down the side of the boiling tube, holding the boiling tube at an angle. DNA is insoluble in freezer-cold ethanol and precipitates or comes out of solution at the boundary of the 2 layers. The DNA is now visible. We can then gently lift the DNA strands out of the boiling tube by twisting a glass rod in the ethanol. And the result is that the extracted DNA appears as a white, stringy mucus.